Hi, my name is Mark, and today we'll share our work on Polyphony, an interactive transfer learning framework for single-cell data analysis. Single-cell RNA sequencing quantifies the transcripts per gene present in individual cells, resulting in a cell-by-gene expression matrix. Ideally, we want to use this data to assign cells to cell types. Historically, cell type annotation has been a time-consuming manual process in which biologists have used prior knowledge of cell type marker genes. One way to reduce the time and knowledge required to perform cell type annotation is to use a reference-based approach. We might envision simply training a supervised machine learning model on the reference dataset and then using this model to predict labels for a query dataset. Unfortunately, the technical variation that exists between sequencing experiments complicates things. We want to remove this technical variation, but it's entangled with the biological variation that we want to preserve. There are several existing workflows for reference-based annotation. A common approach is to create a joint embedding of the reference and queries datasets and then map the query cells onto the most similar reference cluster. However, the question of how to measure similarity between cell groups remains. An existing approach is to use distance in the latent space of the joint embedding as the similarity measure. However, this does not exactly reflect how biologists would compare two groups of cells. Biologists would consider the genes that are differentially expressed between the two groups and then look to external resources for validation. To better understand the limitations of existing reference-based annotation workflows, we interviewed 7 experts in single-cell data analysis. From these interviews, we learned some key insights, including the fact that data integration algorithms are developed under different assumptions, that experts expect to check the annotation results after each data processing step, and that experts want to inspect gene expression profiles before adopting annotation results. Based on these insights, we developed a method to incorporate expert knowledge in an interactive and iterative integration process. Specifically, we use the concept of anchors, which we define as analogous cell populations across datasets, as a mechanism for communication between the human user and the reference-based annotation model. For example, this is one anchor consisting of the set of green cells from the reference and this cluster of gray cells from the query, which the model considers analogous given the current state of the data integration. This is a second anchor consisting of these orange cells from the reference and these gray cells from the query. Anchors are useful because they reveal how the model intends to remove batch effects and because humans can operate on the anchors to guide the model towards more reasonable corrections. In this work, we contribute anchors as a paradigm for communication between users and the annotation model, and we contribute an interactive visualization system which facilitates human-model collaboration. We call our interactive transfer learning framework Polyphony. In the following slides, we'll describe how it works. In the first step, the system recommends anchors to users by building reference clusters, and then assigning the most plausible query cells to those reference clusters. In the second step, users provide feedback to the model about the recommended anchors by using the Polyphony interface. Users can refine an anchor by editing the set of query cells assigned to the corresponding reference set. They can confirm an anchor, or they can reject an anchor. Users can also add new anchors by selecting a set of query cells. In the third step, the system fine-tunes the model according to the confirmed anchors, where the goal is to simultaneously minimize the original loss and minimize the distances between the confirmed anchors and their corresponding clusters. After fine-tuning the model, the embedding of all query cells will be updated. We designed interactive visualizations both to present the current anchors and embedding space to the users for inspection, as well as to enable users to interact with the model and provide feedback. These designs have been informed by our interviews with experts in single-cell data analysis. To allow high-level comparison between the reference and query datasets, we first project the cells from the latent space into the 2D plane using UMAP, since it is a familiar way for biologists to make sense of large single-cell RNA sequencing datasets. The color of each cell encodes either the cell type or the predicted cell type. To enable visualization of very large reference datasets, we use contour lines with a pattern area to encode the reference cell density. This also facilitates distinguishing the reference from the query dataset and overlaying the two on the same scatter plot. To provide users with a better understanding of the integration quality, we developed a glyph to encode the distance between the reference and the query cells included in each anchor. The width of the outer gray line encodes this distance. This visualization design enables a dataset level understanding. Users can gain an overview of the joint embedding space, identify cell populations of interest, and understand how the model is mapping clusters of query cells onto corresponding cells in the reference. We also want users to gain an understanding at the level of smaller cell populations. In an anchor set view, users are presented with the current list of anchors in a table. In the marker view, users can focus on a particular anchor of interest and investigate that anchor at the gene level. The three columns show the top differentially expressed genes that are shared in the query and the reference, or are differentially expressed in only one of the two. The horizontal bars display the z-score of the differential expression results, and the triangle glyphs encode the ranking of each gene among the top differentially expressed genes in the query and the reference. In the Polyphony workflow, we have joined the integration process with the understanding process. We found that anchor-based methods leverage data correspondence for integration, while integration helps experts understand the data correspondence in an interactive and complementary way. In Polyphony, we support visual summaries of and interactions upon anchors, making the anchor-based integration process more transparent. The concept of anchors is effective in part because case-based reasoning is a common human reasoning procedure. Following from this, we find that the concept of anchors is well understood by experts, and experts also understand how to provide feedback using anchors. To learn more, please visit our long presentation at BioVis, which contains a demo, use case, and evaluation results. For more information, check out our preprint or our repositories on GitHub. Thank you.